So suddenly the problem of rapid osmosis, transmembrane water permeability became clear. There was a new protein which we named aquaporin, now known as aquaporin-1, first molecular water channel, basically the plumbing system of the cells. Now, being a small lab, really we were a very small lab, we were not a rich lab, we had small number of grants. We needed to team up with others, and this is where science is such a rich community. We were able to team up with a number of experts from around the world. I'll show you some of the pictures to do a number of important steps which we could not ourselves do. First, we teamed up with these two gentlemen, Yoshinori Fujiyoshi from Kyoto, Japan, and Andreas Engel from Biozentrum, University of Basel in Switzerland. They're experts in the technique of membrane crystallography. And in short, we saw this structure. If you look on the left panel, from above the cell, the water channel is a narrow pore through the protein, 3 angstroms in diameter. And I think many of the students will realize the average diameter of a water molecule, 2.8 angstroms, is just big enough. And viewed in cross-section on the right, there's a single channel connecting the outer surface of the cell with the inner surface of the cell, single aqueous pore, allowing water to move with almost no resistance. And there are barriers to the movement of other solutes. For example, protons, the hydronium ion, the positively charged domains repel it. So here we have water in bulk solution, free exchange of protons. Here, water in bulk solution, but this narrow pore, 20 angstroms in diameter, single file movement of water and water alone. And this was work of David Kozanol, a talented student in the laboratory who's now faculty member at Harvard University. So if you look at the space filling model of water at the center of the core, you'll see it's surrounded by special residues. Arginine, a positive charge. Histidine, a partial positive charge, compelling proton movement. Cysteine, the mercury of interface. Notice it's big enough for water, just big enough. Now, the work of others has extended this structure, and I'll talk about the other homologs in a moment. We're also interested in the physiology of water channels, as Dean Cocker mentioned. In order to establish a physiology, we had to know where, specifically at a cellular and subcellular level, where the protein was expressed. And we achieved this by teaming up with Søren Nielsen from the University of Aarhus in Denmark. We localized the aquaporin-1 protein in the kidney and the proximal nephron. So shaded in green, here's the glomerulus where filtration occurs. Proximal nephrons descending to the loop of heaven. And each of our kidneys has about 1 million nephron units. And the green highlights the location in the proximal nephron where aquaporin-1 is present. So it's present here, and this is where constitutively high water permeability exists. It's not present in the ascending limb, negligible water transport, and it's not present in the collecting ducts, where regulated water transport is known to occur. Surprising, collecting ducts didn't have it. It was a clear prediction that other homologs must exist in kidney. So 2 microscopic slides. Shown here is a light microscopic preparation of a rat kidney. What you should see, the tubule here, the lumen where the primary urine flows is surrounded by an apical membrane, intensely staining with an antibody for aquaporin-1. Present also in the lateral membranes separating the cells and the basal membranes, and not present in the collecting duct. Now, if you look by immunogold electron microscopy at this apical bush border here, you see infolding of membrane. I think you can see that. It looks like hair-like projections. And the black dots represent immunogold, nanogold particles attached to an antibody to aquaporin-1. So the protein is all present at the absorptive surface, not inside the cell. Ideally poised for maximum absorption into the epithelium. So how does this allow water transport to occur? Physiologists here will be aware that every day our kidneys will filter about 200 liters of plasma, filtering the same plasma again and again. 200 liters of filtrate is then passing through the reticules, and 99% of the water is reabsorbed. Imagine if you made 99 liters of urine while you would last only a few minutes as you were doing the shot. We must efficiently filter and reabsorb the water. But we must reabsorb the water, but not the acid or any waste substances. And the explanation is the distribution of the aquaporin 1. The primary urine flows through the lumen, enters through the apical surface, tight junctions prevent water from slipping between the cells, exits through the basal membrane. Transcellular movement. Almost 200 liters of water per day. It's amazing. Now, as a medical doctor, I'm very interested in the causes of disease. It's our hope that our scientific work will provide something useful to prevent or treat disease, at least the understanding of disease states. And before the human genome was solved, we localized the chromosome for aquaporin 1 in the short arm of chromosome 7, where we had a candidate locus, the Colton blood group antigens. This is work that shows over the transitions in the lab. And in short, a long series of experiments, we confirmed that the Colton blood group antigen was a polymorphism in the outer surface of the aquaporin 1 protein. But we were made aware that in the whole world, only a small number of humans had ever been identified lacking the Colton antigens. Well, obviously, we were very interested in these Colton null people. And because of the wonderful collaboration amongst blood group referencing laboratories, particularly the National Center in Bristol, England, we gained access to a number of Colton null individuals and confirmed that they had mutations in the gene encoding aquaporin 1. Our thought was, well, this protein must be so important that we severely disabled. In fact, the aquaporin 1 null individuals all felt well and looked well. They didn't feel sick at all. Here's a photograph of one. With her permission, we took a picture of a lady from south of France who's a Colton 1 null individual, an aquaporin 1 null homozygote, with two of our colleagues at Johns Hopkins. She looks well and feels well. And most of the time, all of us look well and feel well. But when we're stressed, certain mutations become very clearly manifest. For example, I suffer from a mild form of asthma. I'm around the laboratory animals, I wheeze. But by using a corticosteroid inhaler and staying away from lab animals, I'm fine. Others may be sensitive to salt in the diet. Too much salt can become hypertensive. So silent carrier state is very common. When stressed, we expect this will be a significant phenotype. And Landon King, a young man shown here, did a complete renal tubule analysis of these people and found that they had normal renal function except when thirsty. Every night, we're lucky we sleep 7 or 8 hours. We drink no food. In the morning when we wake and we've been thirsty 7 or 8 hours. And our morning urine, of course, is the entire night's collection. But the next few drops of urine are highly concentrated. So normal individuals will concentrate the urine from physiologic chemicity, 280 milliosmolar, up to about 1,000 milliosmolar overnight thirsty. The aquaporin null, one null individuals can concentrate a little bit, enough to get to the night, but they would surely get into clinical trouble with dehydration long before any of us would. So it's a pretty important phenotype. Landon, also an outstanding physiologist, just in lung, demonstrated the presence of the aquaporin 1 protein in the capillaries of lung. And here's a human capillary. You should see the immunoglobulin decoration at both the top and the bottom of both surfaces. And in a collaboration at Johns Hopkins, he did some physiological testing of water permeability in lung. Basically, images were taken of the deep lung structures using a high-resolution CT methodology developed at Johns Hopkins about 30 years ago by a young scientist who came to Johns Hopkins from Algeria. His name is Elias Serhouni. He became the director of the National Institutes of Health and now is the vice president for research at Sanofi in France. Young scientists make discoveries which are important. And using this Serhouni technique, we studied the structures. And so this is a bronchiole and lung. Notice the thin walls. So here's an adjacent venial. Here are the same structures in a normal individual after infusion of 3 liters of physiological saline. what you should see is that the venial has become ignored. And that's found in all normal and aquiform null individuals. But the release of fluids in the venial, the surrounding space around the bronchiole, notice the thickness is increased dramatically. Found in all normal individuals, not found in aquiform null individuals, indicate there's a delay in fluid release from the vasculature and the blood vessels in the lung, which we think may be of large physiological importance at the time of birth when we reabsorb massive quantities of amniotic fluid. So by homology cloning, our laboratory and several other laboratories jumped in and cloned out now hundreds of different aquiforms. Basically every life form has at least one, if not multiple aquiforms. And shown in this disease, this gene pileup, are the two subsets of the aquiforms from the mammalian or from the human repertoire. At the top shaded in blue are those which we call classic aquiforms, water channels. And yellow at the bottom are those which are permeated by water plus glycerol, which we term aquaglyceriforms, closely related but not identical. And here's HTP1, our original discovery. And I'll talk about a few of these others just to illustrate the diversity of expression, physiological function, and what we believe to be the pathological significance. So when I talk about the proximal nephron, I mentioned the collecting duct does not have aquaporin 1. Prediction produced sort of a foot race among scientists to clone the homological collecting duct. The vasopressin regulated water channel collecting duct is known to exist in the collecting duct. And obviously, I'm interested in this. And the answer is that a homolog, known as aquaporin 2, similar but not identical to aquaporin 1, a separate gene, is present in renal principal cells in submembranous vacuoles, vesicles, in a basal state, but it is transported to the cell surface by vasopressin. So vasopressin, I won't go through the biology here, but goes through a receptor leads to a phosphorylation and exocytosis, so water permeability occurs. But look, the lateral membrane does not contain aquaporin 2, it contains aquaporin 3 or aquaporin 4, so it's in concert, these work together. Now, clinically, these are very important. Long before modern physiology, primitive tribal people recognized that drinking large volumes of fluid led to increased urine output. Thirsting caused decreased urine flow. The Plains Indians in North America could wage war parties in the middle of the night by warriors drinking large volumes of fluid at sundown. They'd get up to 3 hours, 2 hours to void their urine.